Hello everybody, good evening and welcome to your very own BYJU'S 9th and 10th grade channel. I'm your teacher Aishwarya and I welcome you to today's class where we're going to be kick-starting your preparation for your periodic test that I know it's going to be upcoming. But of course students understand that this preparation is not just for your periodic test but for all the tests that are going to be there in the future. This is going to be super applicable for any of you who watch it at any point of time, because in today's class, we are going to be covering super 30 objective questions or multiple choice questions from a very important chapter, that is the fundamental unit of life. Now in science, of course, when I just take the biology chapters, you have only 3 chapters, which is fundamental unit of life, tissues, and then we have improvement in food resources. And fundamental unit of life is a very important chapter that is there. So everybody, this particular video is going to be super important for you. Now of course, as this video is being live streamed, I can see that a lot of you are here in my live class. But if there's somebody watching this video much later, you can skip a little bit ahead to the part where I ask you the questions, right? So I think Junior World, you are saying, ma'am, the PDFs are not being provided. As we all know, we are working on the back end of it, but don't worry, from now on, teachers are going to be making sure that it comes your way, right? It's our responsibility. So we are going to make sure that it comes your way. So can we please increase the font size a little bit more, like make it a little bigger? Yes, good evening everyone, good evening. I can see a lot of my regular students are here and a lot of my students who are of course pretty new, right? So now many of you are asking me, ma'am, will we only look at objective questions? Will we look at subjective questions? What is the thing? So first and foremost, one set of objective and subjective questions have already been covered on the channel. We'll be tagging the videos of those particular videos, in the comments of this particular video, right? But today what of course we are going to be doing? What can you expect in this video? We will do 30 objective questions, right? And listen, next week of course Ankita ma'am will come once again with 30 subjective questions. So this time around with biology, because you have one very huge chapter, maybe unlike in the other cases where you have two chapters, the biology syllabus in science is slightly less, which is why we have to split a little bit, right? And if you are attending my class at any point of time, whether you're a live student, whether you are watching this video later, these are things you need to keep in mind, right? What do you need to have, everybody? Have your notebooks and your textbooks. Yes, please make sure you have your notebooks and your textbooks ready with you. Make sure you have all the stationery, right? Make sure you have your pens, your pencils, because there are going to be some important pointers that I'm going to, be discussing today, which will be helpful on an examination point of view. Then your water bottle, right? So as we all know, I have my water bottle as well. You should have your water bottle and a quiet place to focus. Now, very important thing is when you are studying, right now this is an examination preparation. Don't think of it as I will watch this video and then I will prepare for my exam. Then there is no point. You're not being smart with your time, right? If you are going to use this particular class as a class where you will finish end to end of fundamental unit of life, then you need to make sure that you have a quiet place to focus. And understand that today's class, right? Today's class is going to be for 2 hours, right? So today's class is going to be for 2 hours, and trust me, whoever's watching this video live, I will try my best to make sure that I clarify whatever doubts are there for all of you. And of course, those of you who are having some doubts later on, you can let me know in the comments of this video, right? So students, very quickly, are we all ready for today's class, right? Everybody quickly give me a thumbs up in the live chat to let me know that we are good to go. And for students watching this video much later, if you see, we've done one shot, we've not done a one shot, but we've done chapter summaries, we have done everything in great detail, all the important topics, right? Which is why if you have any doubts, just check the videos out, because I have done in-depth concept explanation. Today is not for me to explain concepts in detail. Your exams are close by, which means how to understand the concept easy is what I will be teaching you. Secondly, I will ensure how you need to make sure that all of you, sort of apply the concepts that you have with you, right? So jaldi everybody, please make sure you share this video with all of your friends. Do not forget to hit the subscribe button on the channel, and very importantly, do not forget to hit the like button on this video, right? I need to have at least 100 likes on this video. So let's get started everybody. It's not going to be a Menti. These are going to be objective questions, and we have different rounds, right? So just to make this a little more interesting for you, we're going to have different rounds, which means we are not going to cover cliche objective questions. We will do assertion reason, we will do diagram based questions, we are going to be doing experiment based questions, practical based questions. We will do a lot of different things, right? So today we may not have YouTube live polls as well because we're a little short on people who may help us with live polls, but don't worry, let's get started. Yes, this is for class 9, round 1. Now round 1 is an easy round, right? I always start round 1 very simple and easy. So these are going to be direct questions, okay? So the first set of questions for you is this. The word cell is derived from what? The Greek word that means small box-like structure, a Latin word which means a little room. Does it mean that a Greek word that means a little room or a Latin word that means small box-like structures? Yes? Cell came from what? Very good everybody, very good, right? So now of course we all know the term cell was coined by who? It was coined by the scientist who was Robert Hooke, right? It was coined by Robert Hooke. Now here it's slightly tricky, right? We know that cell is derived from the word cellular, which means a little room or a little space, right? So many of you here have got the answer. Now, of course, the correct answer here is option B. It is a Latin word, right? It is not a Greek word. Yes? Now, you can say small box-like structure or a little room. But in this case, we know that when Robert Hooke observed it under the microscope, he saw that, you know, they appeared like little rooms of a monastery. But the term cell came from cellular, which meant little room. It's not a Greek word, right? So the correct answer here is option B. Now let's move on to question number 2. Yes? Question number 2 is the term protoplasm, right, is coined by who? Is it Purkinje, Robert Hooke, Virchow, or Robert Brown? As you all know, this right here is not, there are no YouTube polls for all of you, so you need to give me the answer in the live chat, right? Yes, very good, very good. So now here we just discussed Robert Hooke coined the term cell, so it's not Robert Hooke. We know Virchow that was there, he had a very important role to play with respect to cell theory, right? And he was the one who added a point into the cell theory where he said all cells arise from pre-existing cells. So it can't be him. We are left with Purkinje and Robert Brown, right? Now, what did Robert Brown discover? Exactly, very good Ankita. Robert Brown discovered the nucleus, right? So it can't be Robert Brown either, which means it is Purkinje. Additional question for all of you, right? Make a note of this. You can get a 1-mark question, you might get a 2-mark question where for subjective they can ask you to differentiate. So they may ask you to differentiate between cytoplasm and protoplasm, right? So they can ask you a 2-mark question. So everybody tell me what is cytoplasm? Junior World, you will get immediately after the session is over. Okay, my promise to you. Yes? Quickly, what is cytoplasm? I need all of you to tell me in the chat, right? What do we understand by cytoplasm? Protoplasm. Yes, okay. Many of you, okay, I think I said cytoplasm, so you're giving me the answer. We know that cytoplasm is the jelly-like, or it is the viscous fluid that is present where? It is present between the plasma membrane and the nucleus, right? So in the case of eukaryotic cells, it is between the plasma membrane and the nucleus, and we find all the cell organelles that are suspended. But what is protoplasm? Exactly, protoplasm is the living component of the cell, right? So you'll have to write down what all it includes. Yes, it is the living component of the cell, which is why this is the main difference between cytoplasm and protoplasm. Many of you here are still getting confused as to the difference between cytoplasm and protoplasm, which is why I have written this down so that all of you are clear with this, right? Make a note of it, write it down in your textbooks. This will come handy. So let's move on to question number 3. Yes, very good, very good. Question number 3, everybody. The cell wall of the plant cell is made up of what? Very, very easy. Right now, this you can close your, if I say close your eyes and give the answer, you will tell ma'am, how can I do that? But we know that we are talking about which component makes up the cell wall. Glucose, fructose, cellulose or protein, right? This of course is very simple and easy. We know that cell wall of plant cell is made up of cellulose. Quick follow-up question. Cell wall of fungi is made up of what? Very quickly, this is an additional question for all of you, right? Yes, I need all of you to tell me. Now I know some doubts are coming my way, but if you are all comfortable with it, can I take a doubt board section so that in the doubt board section, I'll make sure that I take those doubts, right? Yes, very good. We know that fungi is made up of chitin, or the cell wall in fungi is made up of chitin. And what about in the case of bacteria? What about cell wall in bacteria? What is that made up of? Very quickly, I need the answers coming my way. Yes, I know. Now you will think a little bit, ma'am, bacteria cell wall, exactly, peptidoglycans, or you can say complex polysaccharides. But correct answer is peptidoglycan. Now this is little extra in case if you want to do Olympiads and all that, this will come handy, right? Yes. Moving on. Which among the following is not a function of vacuole? Is it storing metabolic waste temporarily in their body to help with process of cell division, to maintain turgidity, or to provide structural support? Yes? I know. This village of Palampur is making all of you get very nervous. Don't worry about it. I'll be taking that class on Friday. Now, actually, that is something I'm going to deviate from the plan because ideally, I should be doing your periodic test preparation. But because it's there in your periodic test, I will be taking one shot that day. On Friday, I'll be taking that class, okay? So don't worry about it. Yes, very good, very good. We know that which among the following is not a function. Then it has to be the fact that it helps with the process of cell division. Now, which cell organelle is responsible for that in animal cells, right? So let me rephrase it. Which cell organelle is responsible for helping with the process of cell division in the case of animal cells? Yes? Creative lifestyle. If the screen is not clear, I request you to go to settings and increase the resolution. Yes, nucleus, exactly. Centrioles are also involved, right? So nucleus is your control center, but along with that you have centrioles or centrosomes, more or less correct, right? These are the two cell organelles which are involved. Now here we're talking about which is not a function, which means you need to identify which is the function of vacuole. Now see, store metabolic wastes, right? Now when you talk about metabolic waste that is there, we see that you can talk about, I'll rephrase it a little bit to maybe, temporarily for a brief period of time before it gets expelled. They tend to store a little bit of excess water and everything, right? So we see that this right here also helps in maintaining turgidity, and along with that they provide structural support as well because they maintain. It's not the structural support the cell wall provides. This is in terms of maintaining that turgidity, right? Which is why in this case I would suggest the correct answer is option B. Now see, here also I will tell you one thing. This is a question which was there in some of the earlier question papers from some schools. Now here the question is little vague. I will agree, okay? Because toxic metabolic waste and all sounds very fancy, right? But here they are telling with respect to excess water. So try not to, in such cases when the options don't look very vague, right? Look for the most obvious answer, right? Yes. Let's move on to question number 5, right? An unripe green fruit, yes, changes color when it ripens. What exactly goes on? Does this mean that chromoplast becomes chlorophyll? Does it mean chromoplast changes to chromosomes? Chromosomes change to chromoplast. Chloroplast changes to chromoplast. Now see, one thing we know, we're talking about something which was green in color becoming colored, right? So in this particular case, we know that chromosomes per se, right? It's not chromosomes that are getting involved. Although they have a role, not directly. They're not getting converted into chromosomes because of which color is getting imparted. So we need to look at which cell organelle imparts color. Now in this particular case, we know that here it is the plastids that are there. You have chloroplast, chromoplast, leucoplast. Now we know leucoplast does not have any color, which means in this case the correct answer is option D. Chloroplast changes to chromoplast, right? Now see here it is saying chromoplast changes into chlorophyll. Chromoplast is an organelle. Chlorophyll is a pigment present inside a cell organelle, right? So this also would not be the correct answer. Yes? Utkrisht Bacha, I will take your doubt. So just before I proceed with round 2, I'm going to stop and take a doubt board, okay? But let's get started a little bit because even your time, right, matters. Now when I say change, right, does not mean that your chromoplast that is there will suddenly, become, it'll destroy and it'll become something else. No. So basically when an unripe fruit, right, so when you have a unripe fruit, yes, and you are saying that it becomes colored. Basically what happens is that when an unripe fruit is green, it is green in color due to the presence of chlorophyll, right? And technically when you think about it, right? So when you really think about it, your chloroplast that is there, right? Isn't chloroplast a type of chromoplast? Will you all agree with me? Yes or no? Chloroplast is actually a type of chromoplast. Yes or no? I want you to tell me. How do we define a chromoplast? It is a plastid which has pigment, right? Yes, no, yes, no. Actually, as a matter of fact, you can say that it is a type of chromoplast, but we kind of call it out separately because chloroplast function, because the functioning of chlorophyll is very, very important, right? Which is why we say that chloroplast is something we call out separately. But chromoplast is a plastid with pigment. So chloroplast is technically chromoplast. Yes. Now, secondly, in this case, what happens is that when the color starts forming, basically what happens is that there is synthesis of other pigments, right? So we see that other pigments slowly start to get synthesized within these plastids, which is why in this case, yeah, it has carotene, some chromoplast has anthocyanins. You have different kinds of pigments, right? So that's something that you need to understand, all right? Okay, moving on to question number 6. Now question number 6 is an NCERT exemplar question, which I think if you've attended my class regularly, this is something that I have already taught you. A cell will swell up if you have 4 options from which you need to identify the correct answer, right? Yes. Amanda, ma'am, I don't understand what ma'am said a little bit also in school. But have you watched some of the sessions, right? So in case if you watch the class carefully, I'm telling you it's going to be helpful. But whatever you have not understood, please go back and watch the one shot, right? Put it in 2x and watch it. And then you come and watch these classes. It will be really helpful, right? Now if you don't understand anything and you watch my class, it might be a little tough for you. Okay, when will cell swell? I always tell you, cell will swell in a hypotonic solution, right? So which means that the concentration of water molecules in the surrounding medium should be more than the cell, so that water can enter into the cell, which is why in this particular case, we see that the correct answer here is option number B. Option A says that concentration of water molecules in cell is higher. If it is more in the cell and less in the surrounding, water will move outside through osmosis. Now, if water molecules are the same, then there will be no net movement of water. Concentration of water molecules does not matter. That also doesn't make any sense, right? Which is why you can eliminate out your 3 options, right? So, we are clear with this concept. Are we all clear? Yes or no? Osmosis in virus. See, virus, as you can see, they have a protein encapsulation, right? And for them, it works quite differently. So, maybe not, right? Yes. Next one. Digestive enzymes are found where? Is it found in mitochondria? Is it found in cell membrane? Is it found in ribosomes? Or is it found in lysosomes? Easy peasy. The organelle which is responsible for getting rid of things will have digestive enzymes, right? Which is why in this particular case, as most of you have told me, correct answer here is option D, which is lysosome. Now this I'm not going to spend too much time because we already know the answer to this. Now let's move on. Now first few questions are very easy. After that the tough ones are coming, right? It's always, we need to feel good in the beginning. Now, an undefined nuclear region of prokaryotes is known as what? Nucleus, nucleoid, nucleolus or nucleic acid. Now we know that in the case of prokaryotes, we know that they do not have a well-defined nucleus, which means that their genetic material is found concentrated in a particular region in the cytoplasm. And we call this region as the nucleoid region, right? Which is why the correct answer here is option B. Hello, intelligent boy. Hello. Okay, for students who are telling me, ma'am, my screen is blurred, I request you to increase the resolution, go to settings, advanced settings, put it in 720p, you will be able to watch it, right? Ma'am, nucleoid is made up of nucleic acid. Good observation, right? But understand that we're talking about the undefined nuclear region, right? So the region does not only consist of genetic material, it's a whole region that you see in and around it, right? So we see that it's a suspended region, which is why we call it as nucleoid region. Yes? Okay. Moving on to question number 9. Which among the following has its own DNA? Vacuole, mitochondria, Golgi, ribosome. So easy, right? Very, very easy. Yes, everybody's saying ma'am B is winning with runs. Amazing. Very good, very good. Yes, exactly. Easy peasy, lemon squeezy. Somebody said ma'am ribosome has its own DNA. See, ribosome is responsible for protein synthesis, right? So technically ribosomes are found within the cell organelle, but in this case the correct answer is option B, which is mitochondria, right? Which is the other cell organelle, which is semi-autonomous, or I can say that this also is an organelle which has its own DNA. Exactly, Avni. Very good. Plastids also have their own DNA. So this could be a 2-mark question which they ask you. List or write down any 2 organelles which have their own DNA, or they may ask you which are semi-autonomous in nature. That means that in this case the correct answer is these 2, right? Plastids and in the case of mitochondria. Now let's move on to question number 10, after which I think we have a small break where I can take some doubts. While observing a human cheek cell under a microscope, what will the student not see? Will the student be able to see nucleolus? Will the student be able to see mitochondria, nucleus, chloroplast? Now understand that this particular question is not about what you will really see under the microscope. Good. I know that now many of you did not unnecessarily get confused, right? Yes, very good. The correct answer is option D, chloroplast. Now, can any of you tell me why? Why is the answer chloroplast? Yeah, you can say ribosome. We normally categorize ribosome as a cell organelle because it kind of does an important function, right? So we do say it's a cell organelle. Exactly. Anupama's answer is the first, along with Abhimanyu, Harsimran, Dhriti. Exactly, right? See, many students, when you read the option, no, you will first think about what you will really see under the microscope, because the question is twisted in a little way, saying that while observing a human cheek cell under a microscope, what will the student not observe? So some students will think, well, what can I literally not see under the microscope? But at this point, what they're really asking you is, what are the cell organelles you will see in an animal cell versus what you will not see in an animal cell, right? Now, will you observe a chloroplast in any animal cell? No, right? At this point, we know that chloroplast is something that we find only in plant cells, right? So at this point, irrespective of whether you have electron microscope, you have crazy, imaging, whatever it is there, photo imaging microscope, whatever you find, right? You can see nucleolus, mitochondria and nucleus, but you may never see a chloroplast because that's something we find in plant cells, right? So with this, everybody, we are done with 10 questions, and I'm very proud to say all of you have scored 10 out of 10, right? Which means that deserves 3 stars for all of you, right? So very happy and very proud of all of you. Yes? This is a very popular question, and this question right here can come, right? Now, before I go ahead, I'm going to take 5-10 minutes. Yes, I'm going to take 5-10 minutes to clarify some of the doubts I saw in the chat, right? Yes. Very quickly, everybody, the chat has frozen. 1 second. Let me just open this in my phone. Okay. Where does digestion occur? Right? All right. Yes. Doubt in ma'am, why life is called a protoplasm? No. Protoplasm is made up of all the living components that are there. And I think Harsimran asked me, ma'am, is cell membrane part of it? See, let me tell you one thing about the cell, which I find that it is a little tricky for all of you to understand, right? Is that individually these components that are there, right, when you think about it, individually when you think about these components, they all appear to be non-living because they are all either carbohydrate, proteins, fats, and you're like, ma'am, how can I perceive it to be living? But normally, even the cell membrane, we incorporate it as a living part, or it's actually somewhere between living, non-living, all right? Like it has like a weird definition, to be honest, because there's a lot of research which is still being done on the cell membrane, but normally we incorporate it as a part of the protoplasm as well, right? And here at this point, what you think about it is that all these come together and make or provide that kind of life. We are able to survive because of all these things coming together and working together, which is why we say that the protoplasm is the living component, right? Now, next up is, ma'am, which part of the cell produces lysosomes? We know that lysosomes that are there is produced by the Golgi apparatus. But I hope I answered your question, Harsimran. I hope I did answer your question. All right. Now, next up, what is semi-autonomous? Which means that when you say semi-autonomous, autonomous means it functions on its own. Semi means it's not entirely functioning on its own, but partially. So, in the case of mitochondria, in the case of chloroplast, we know that although they have their own DNA, right? We see that certain proteins and things that they require, they produce by themselves, and they're not dependent on the nucleus for all of that. Yes? Okay. Ma'am, osmosis diffusion. See, all of you should have gone and watched that particular video, right? So I hope that all of you have watched it. Now I can see that so many doubts in this particular thing. Endocrine system later, 10th standard, please, let's not get it. Osmosis, right? What is osmosis? We know that osmosis is simply said as diffusion of water, right? But here semi-permeable membrane is involved, which means osmosis is the movement of water molecules from a region of its higher concentration to a region of its lower concentration across a semi-permeable membrane, right? So it's across a semi-permeable membrane. Now understand that when you say endosmosis, endo means enter. That means water is entering inside the cell, right? That is endoosmosis or endosmosis. Now exoosmosis, exo means exit. That means water is exiting the cell through exosmosis. And we know that that is what? So it goes outside the cell through the process of osmosis, right? So during endosmosis, because water is entering inside the cell, it will swell. Yes, in exo, exit, that means it is going to shrink here. Yes? Ma'am, how many layers of plasma membrane is present? Good question. So actually in the case of plasma membrane, right, in the case of prokaryotes, they have something called as LPS, okay, lipid polysaccharide, if I'm able to remember it right. Yeah, they have something called as an LPS. Thickness varies, right? Thickness varies across different kinds, right? There are, in some cases, we see that the LPS might be a little thicker, a little thinner, difference from type of bacteria. Cell division meaning, cell division is nothing but when a single cell, right, it undergoes splitting in such a way that all the cellular content is divided between the two cells, right? Now, this can happen as equational division or it can happen as reductional division. Yes, RER, SER, I have questions coming, I will explain that, right? Mitosis is equational division, right? Mitosis is equational division where you see that the parent cell will give rise to two daughter cells which have the same equal number of chromosomes. Meiosis is reductional division where they have only half the number of chromosomes, right? Ma'am, is protoplasm the cell itself? See, there are some non-living components, right? So the cell wall that is there is said to be a non-living component. So sometimes protoplasm primarily excludes the living part. Yes? Ma'am is not ignoring anybody, trying to read as many as possible. Sap vacuole and contractile vacuole. See, contractile vacuoles that are there, right? Sap vacuoles basically store sap, like in the case of plants. They store sap. So it will include water, it will include some, proteins or some other components like some carbohydrates. It will include all of that. Contractile vacuole you will see in amoeba, right? Which will store water and will also help in the movement. So that's contractile vacuole. Phagocytosis. Amoeba, eating food, engulfing, that is what you call as phagocytosis, right? Yes, exactly. What is DNA? Genetic material. Why is virus an exception to cell theory? Simply because viruses cannot make more of themselves on their own. They're dependent on the host cell, right? They are not able to survive independently, which is why we see that they do not fall under the category of this one. What is that? They don't fall under the category of cells. Okay, students, I am going to take 1 minute of time now. See, I'm going to keep taking a lot of doubt boards, okay? But understand that I also have a lot of questions that's going to help you out with the same. So shall I move on to the next set of questions? Then I'll pause for doubt board. And whatever questions that I did not answer, I will pick it up then. Yes? Okay. Shall we get started? Shall I proceed to the next set of questions? Otherwise, today's class will be easily 5 hours. All of your questions, I will keep taking it. Physical features. Bacha, you want Palampur first or you want physical features? You tell me that in the live chat, okay? But see, social studies separately, bio separately. I have questions on Golgi body, I have questions on ER, and then I will teach you that. Pass active transport also, I will take great action, but you keep a happy face and you ask me. No, I love when people send me happy smileys on the chat rather than angry face ones. Yes? Okay. Now don't get distracted with social science. Let's get started with round 2. Yes? I will take your question, Bacha. I have a question on the same also. Plasmolysis also I have a question. Don't worry. Turgidity, flaccidity, everything I have questions. So I will teach you more when I have that question coming my way. Okay? Let's move on to round 2, which is going to be diagram-based questions. Now let me tell you, in biology, diagram-based questions are going to be very common. If you are somebody who wants to do 11th and 12th in biology, or you want to pursue bio in 11th and 12th, start getting a little more comfortable, right? Yes. So now everyone, I need you to focus on this particular diagram, which is of the bacterial cell. And notice how there are 3 parts which are mentioned here. You have part 1, part 3 and part 2 that is mentioned. Okay. Now, very simple, easy question. Identify the given label diagram and then match or identify the correct option, right? So now what you need to do, identify what is 1, 2 and 3. Now you have 1, you have 2, you have 3. Now 2 is very obvious. 2 is nucleoid, okay? 2 is nucleoid because it's bacterial cell, which means that we know at this point bacterial cells are prokaryotes. They don't have a well-defined nucleus, right? Now just with option number 2, you will get the answer. Because if you see in option A, 2 says cell wall. Option B, 2 is nucleoid. 2 in option C is nucleus, while 2 is cytoplasm. With this very clearly, you can eliminate out A, B, A, C and D, right? Now this is of course small techniques, but that does not mean that you apply this. You should also be able to identify 1 and 2. I mean 1 and 3. Now in this particular case, what is 1? We see that 1 right here is nothing but the cell wall of the bacterial cell, and 3 right here is nothing but the plasma membrane, right? Which is why in this particular case the correct answer here is option number B. Yes. Okay. Just give me one moment, everyone. Okay, all right. Very good, all of you. Very, very good. So see, these are some small techniques also which will help you solve these objective questions, right? Now let's go on to question number 12. Okay, we have 30 questions. We are still in question number 12. Yes. Ma'am, answer in first question is wrong. But I gave you the correct answer, right? Yes. It is very cold in Bangalore. That is why I am sniffling and I am wearing a huge sweater. See, cell membrane, plasma membrane, don't get confused. It's more or less the same thing, right? Plasma we use because it more has like that fluid kind of nature, which is why we use fluid, you know, the term plasma. Now, identify X and Y. Now, everybody has told me, ma'am, X is nucleolus. Okay. You have to identify what is X is nucleolus as per everybody, and why they are saying. Very good. Chromatin fibers or is it chromosomes? I have a question for all of you. Chromatin fibers or is it chromosomes? What is it? Is it A, chromatin? Is it B, chromatid? Is it C, chromosome? What is it? A, B or C? I will look at majority here. Very good, everybody. Very, very good. Right? Now we know that DNA that is there, right? The genetic material, it exists as a double helical structure. It's very, very long, and if you need to fit all this DNA inside the nucleus of the cell, it needs to get condensed, right? Which is why the DNA will first wind around some proteins, right? And we call those as nucleosomes. And then of course it will further condense and it will form thread-like structures called as chromatin material or chromatin fibers. Now when this further condenses, they will form, these rod-like structures that you see, which is called as chromosomes. And we observe the term chromatid during cell division, right? Which is why in this particular case, what is the correct answer? X is nucleolus, right? They are pointing towards, I hope I wrote nucleolus here, exactly. So here, as you can see, right, they are pointing towards this particular part, this darkened structure. They are not marking this. If this part Z was marked, what will you write? If you mark, if I had an extra one which says Z, what will you write that as? If I mark the part Z, what will you identify this as? Can you tell me? Cell membrane. Is it cell membrane? Nucleus, or I'll accept nuclear membrane also. But it's the nucleus, right? Yes. Ma'am, you wrote chromatid instead of chromatin. No, I said there's chromatid also. Chromatin is there, chromatid is also there, and that's what I said. Chromatid is a structure found during cell division, right? So this particular part, if I identify, this will be nuclear membrane, and this whole structure is the nucleus, which is why inside structure X is nucleolus. Very good. I'm so proud of all of you. Now we have a very interesting question here. Very, very interesting question. Identify X and Y and then choose the correct answer. Now, first and foremost, can you tell me which cell organelle this is? Take a minute and tell me which cell organelle are you able to see right here? Yes? Golgi body, mitochondria, plastid. Okay. Mitochondria, suddenly mitochondria has taken the lead. All right. Golgi. See, okay, for people telling me plastid, fine. Golgi is not that. Golgi will look something like this. It will be flattened, right? So this right here is the mitochondria, okay? This is mitochondria. And why is it mitochondria? Whenever you see me drawing mitochondria, remember I draw a lot of finger-like things coming here. So as you can see, this is mitochondria. If it was plastids, it would look something like this, where you will see ladders, okay? There'll be ladders and they'll be connected something like this. Yes, so that is what we understand as or this is how the, chloroplast would look like. It is sure mitochondria. Ankit is right. Now you need to identify which is X and Y. Now X right here is pointing towards some finger-like structures. So what is it? Is it cristae, right? Is it cristae or is it cisterns? Two things you will get confused with. Is it cristae or is it cisterns? A or B? Very quickly. Cristae or cisterns? Exactly. Now, lot of unique spellings of cristae is coming in my way. Spelling, guys, in biology, you will lose marks. You will know every content, but if spelling goes wrong, it will mean something else and then gone case. 1 mark gone there only unnecessarily, right? So here, of course, X right here is nothing but it is the cristae. Where will you find cisterns? Can you tell me? In which cell organelle are you going to find cisterns? Quickly, in the live chat. Very good, Atiya. Very good. We find or we see this in the Golgi apparatus, which I'm using short form and writing GA, right? So if my finger-like projections are called as cisterns, then what is Y? Y is pointing to all that space in between, right? So here, if I'm going back and drawing, this is X. But this part in the center is what we say as Y. Yes? So what do we mean by, what is Y here? Very good, Mohil. Exactly. Y here is the matrix or the space in between where we find the ribosomes, where we'll find the genetic material, right? So that is what is Y. But that's not the question. The question here is, you need to identify the correct answer. So this is your question. See, cristae is the inner, so basically for those of you who don't know what is cristae, right? We know that mitochondria is a double membraned organelle which has an outer membrane and an inner membrane. Now these inner membranes are made into finger-like projections, right? They are made into finger-like projections called as cristae. But what is the function is what you need to identify. A says that function of X is to increase surface area for ATP synthesis. Function of Y is increase surface area for ATP synthesis. Function X is increase surface area for protein synthesis, or Y is increase surface area for protein synthesis. Now see, you know that X has not, what is the main function of the mitochondria? ATP synthesis, right? So at this point we can eliminate out C and D. Now it's not that protein synthesis, see, they make their own proteins, but there's no increased surface area for more and more proteins to be produced. They make proteins what they require. Now what facilitates and makes sure that enough ATP or energy is produced? It is the function of the cristae, right? So what is ATP? Again, I know many of you will ask me. It is nothing but adenosine triphosphate, right? It is the energy currency of the cell. And because mitochondria's main function is to produce ATP, we know that we call it as the powerhouse of the cell because they produce all the ATP molecules that are needed for the whole cell, which means that the cristae are there have these finger-like projections so that they can increase the surface area for ATP synthesis, right? So correct answer here is option A. Now you understand that this right here are two things. Now quickly for all of you, once again I am explaining mitochondria, right? Now mitochondria, that is, there is a double membrane structure. Yes, it is a double membrane structure. So we see that it has an outer membrane and we see that it has an inner membrane, right? Now the inner membrane that is there, I'm just going to make a simple diagram. The inner membrane is made into, this is the inner membrane. And what do we see about the inner membrane? We see that the inner membrane that is there is made into folds, or they are made into finger-like projections, which we call as cristae. Yes? Now what do they do? On the surface of this membrane is where your ATP molecules are produced, right? So we see that ATP, the energy currency that is there, right? The energy currency that is there is produced here. Now in this, what do you say, in the matrix that is there, this part is the matrix. Here you find your DNA and everything, right? We know that they have their own DNA. So we find DNA, ribosomes, and you know the basic functioning happens here. Yes? It breaks down glucose. Very good. Exactly. Are we all clear? Ribosomes you find here. Yes? Good to go? Shall we move on? Everybody, are we ready to move on? Yes. Let's go on to question number 14, right? Let's move on to question number 14. Which one of the following, right? Yes, Ananya, where does digestion happen? Digestion that is there happens where basically your lysosome is what is responsible for digesting or engulfing whatever is not needed, right? Yes. Ma'am, why will mitochondria have their ribosomes? Because see, mitochondria and plastids, they have their DNA and they have some ribosomes. So they're able to synthesize their own proteins. And the reason being is a long story about endosymbiont theory, which of course is something that we cover at a foundation level. But I'll tell you towards the end. Now, which one of the following takes part, this is question number 14, which one of the following takes part in storage, modification and packaging of various chemicals? So you have X, Y, Z and W. Now identify the organelle which is responsible for this. Which organelle is responsible for storage, modification, packaging of chemicals? Exactly, it is the Golgi apparatus, right? Now many of you wanted to know about Golgi. Now Golgi, that is there. So students, just bear with me for a minute, right? Now when you talk about Golgi, right, we know that Golgi is made up of various flattened sac-like structures, right? And what do we call these flattened sac-like structures? Yes, what do we call them? What is it? There are many flattened structures that they are made up of, right? And we call this as cristae, right? So we call, cisterns. Oops, I only made the mistake. So we know that they are made up of, or we call them as cisterns, right? Now we know that the size of the cistern varies, right? So the closer they are to the nucleus, we know that this part of the Golgi will receive, all the materials which are coming in from the endoplasmic reticulum. And as it moves through the Golgi, we see that it will undergo some modification. And finally, in the form of small sacs, like they're like small vehicles, okay? So we see that in small vehicles, we see that various modified proteins, various some amount of complex carbohydrates. We see that at this point, right, all of these things, we know that they are transported through these vehicles to different parts of the cell. And we call these vehicles as vesicles, right? And we also know that Golgi is involved in the production of lysosomes. So on your examination front, you only need to remember these major points. Cis phase, trans phase will not come in exam. I just taught you a little bit extra. Now many of you asked me, see, you just need to look for the flattened sac-like structures. That is more than enough. Ma'am, what are the structures that we represent on top of Golgi as dots? Those are the vesicles, Harsimran. Those dots that you draw, they are basically the vesicles which are there that are coming out. And lumen is the gap. Very good. So now which part it is? You have Z, Y, X, W. Now we know that in this case the correct answer here is option number D, which is W. Okay. Now many of you are asking me, see, if you go back to the diagram, Z and Y here more or less, see, probably one of it is trying to reference lysosome and the other one is trying to reference maybe some ribosomes, right? And X right here is nothing but the vacuole. So correct answer here is option D. Yes? Ma'am, vacuole and vesicle. Vesicles are transport. They are small structures which are produced or they are synthesized by the Golgi so that the proteins or the carbohydrates, whatever they synthesize, can be transported. Vacuole is a huge storage sac which is present. In the case of plant cells, they are big. In the case of animal cells, they are smaller, right? So, Soumya, I hope I've clarified your doubt. Ansuya Bacha, we're talking about Golgi bodies, which whose main function is storage, modification, and packaging. You can just rewind back. Ma'am, cytoplasm is all the things present inside the cell except the nucleus. Cytoplasm is basically, see, if you have cell, you have nucleus. All this jelly-like thing which is present between plasma membrane and nucleus is your cytoplasm. Okay? Yes. Very good, very good. Now let's go on to question number 15. Identify A and B. Look at the diagram. Right, everybody, please look at the diagram. I want you to pay attention to the diagram and see what is happening right here. Yes? Look at these two diagrams. Have you all observed? Okay? Have you all observed carefully? Now, this is a slightly important one. Everybody's telling me, Mom, onion cells. Onion cell and cheek cell. Asta says plasmolysis. Shrinking. Plant cell and plasmolysed cell. Plant and animal cells, okay, many of you are hypotonic. Animal cell, plant cell. Okay. Many of you who identified it with respect to, right? As you all know, right? When you talk about this particular thing, the cells are the same. Understand? Let's go back and observe, right? It's okay if you've gone wrong here and there. Observe the cells. You're saying A cells and B cells. But if you look at it structurally, they are more or less the same. Now, cheek cells that are there, and you will learn about this in the next chapter that I take, which is tissues. Cheek cells are made up of flattened cells, okay, and we call them as squamous epithelium. So when we draw cheek cells, we will draw it something like this, right? So they are flattened cells. Now this hexagonal kind of cells you see, right? Hexagonal or polygonal cells are mainly plant cells. So in both cases, these are your plant cells. But they have undergone a certain process, right? So here you have A cells and B cells. Now what do you observe? Now many of you were confused about this part, which is why I am going to take a minute and explain, right? So now I am going to draw two cells, okay, just because I can't draw more than this at this point. Now as you all know, in the case of our plant cells, we know that this right here, right? When you look at these cells, you have the cell wall, you have the cell membrane, right? And then of course inside you have the nucleus, the cytoplasm and everything. Now in this particular case, what do you observe? You observe that they have a proper shape, right? So we see that they have a proper shape and they look rigid. Kind of like, this is a very common example I'd like to give. Imagine if I took a balloon and I blew it, right? We see that it has that shape because I filled that air inside it. Yes or no? I took the balloon and I blew it and it's become big and it has this shape because of all the air that has gone inside, right? So the shape is intact at this point. Now for students who did not know what we call this condition, if a cell is able to have this kind of shape, it's because of some amount of water that has entered in, right? So when it has that kind of swollen intact shape, we call it as turgid, right? So what is turgid? Turgid is a condition, right? When you say a cell is turgid, it is a condition where it has water which has entered into it, and we see that it is holding up its shape, right? So it is swollen, you could say that. So what is turgidity? Turgidity is a phenomena or the condition, again you can say. So when we say that a cell has a shape intact, it's because of turgidity. Yes? So for those of you who had doubts on turgidity, I hope all of us are clear with this. You can say, hardness, shape being intact. I'm not able to think of the right word, but more or less, right? This is what we are aiming for. Yes? Okay. Now, what happens is that now there is water. Now imagine I take the same cell and I put it inside a solution, right? I put it inside a solution which has very little water and it has lots of salt, right? We see that this right here has lots of salt in it. Now at this point, right, what's going to happen? I take this one cell here and I'm going to put it inside. What's going to happen? All the water is going to go, right? So which means water that is there, which is mainly there in your cytoplasm, right? See, where is all this water? Mainly it is there in your cytoplasm, right? The cellular component that is there. Now when all this water goes out, it will shrink, right? The whole protoplasmic content that is there, all this will now shrink, right? Now because the cell wall is there, we see that there's a lot of empty space. The cell wall will not shrink. Cell wall is a non-living thing, it is rigid, it is holding its place. But the rest of it has shrunk, okay? Because all the water has gone, it has shrunk. Now in the case of animals, what will happen is, in animal cells, if too much water goes, we see that it will get dehydrated and sometimes it will not be there. Now this condition where a lot of water has gone out in the case of plants and it's become very less, we call this condition as a plasmolysed cell. And this process of water exiting out from the plant cell is what we call as plasmolysis. So for whoever was confused with plasmolysis, are we all clear? See, I'm spending my time and teaching you, right? I would make sure that I hope you understand. Ion gaming. See, I know I don't expect everybody to like biology, but I hope I make it interesting for you, if not the fact that you don't like the subject, right? Yes. Okay. So in this case, what happens? A is turgid, while B here is plasmolysed, right? Which is why in this case, the correct answer here is option A. Harleen Bacha, A is plasmolysed. A is turgid, right? While B is plasmolysed. Okay? Are we clear? Pragati Bacha, what is it that you are not understanding? You tell me right now, because I don't want you to feel like, ma'am, not happening, right? Okay. All right. I'll do this once more. Cell is here, right? You all have cell. Okay? Now we know that the cell that is there has cell wall, cell membrane. I'm drawing plant cell. Okay? Let's help our friends out in the live chat. You have cell wall. Cell wall is made up of cellulose. Yes? This is non-living. We have all established this. Okay? Then of course, next up we have cell membrane. We have cell membrane. Now, cell is looking intact, kind of like a balloon. Let's imagine our cell to be a balloon. Now, balloon can look like this. No, when you buy the balloon from the market, when I buy balloon from market, the balloon will look like this. Weak, no shape, wrinkly, right? That's how it looks like this. But when I inflate it or when I put air into it, we see that it will have a shape. Yes, it will have it will be swollen, and we see that it will have a shape, right? It's because of all the air that I put inside. Now, if a cell looks like this, it is because there is water. Okay, there is water inside the cell, right? And we see that because of it, its shape is intact. Okay, we call this condition as turgid. But if at the same time I get rid of all the water, right? So if I remove the air from my balloon, it will go back to looking shrink and sunken, right? So as we all know at this point, we see that if I put this in a hypertonic solution, yes, if I put it in hypertonic solution where all the water will go, then we see that apart from my cell membrane, everything would have shrunk. Now this particular process where everything shrinks considerably in plant cells, right? We call it as plasmolysis, right? Yes. Are we clear? I hope we are clear. If not, I request you to go back and watch. Turgid is flexible. See, flexible is a very difficult word to use. I'll be honest, okay? Because in the case of turgidity, it exerts pressure on the cell wall. So flexible, I don't know to what extent we can say. Let's move on, everybody. So with this, see, I took some time here because we are done. Now students, we are going to round 3. But before I go to round 3, can I give you all a 1-minute break? Can we do a water break right now? Shall we take a quick water break? See, we have only 15 more questions and they're going to be easy ones, right? So we will take a 1-minute water break together and drink water. Yes? And in the meanwhile, students, on a scale of 1 to 10, right? I want you to tell me how confident you are feeling. Now, in the meanwhile, as you all take a water break, I want to help my other students out saying, ma'am, can you give me a definition for nuclear membrane? Nuclear membrane is the membrane, or it is the outer covering of the nucleus. Now, it is not a continuous membrane, but we see that there are certain pores which are present, which we call as the nuclear pore, right? Sonam, I think I have a question, so I will take that question and I will give you the answer. Now, in the meanwhile, if you are telling me, ma'am, I think 10 out of 10, 9 out of 10, I hope all of you have hit the like button on this video. We should have at least 150 likes, and please make sure that all of you go ahead and share this video with your friends as well, right? Okay, just give me one moment. See, I'm taking time with this particular class because you have exams coming up, okay? And I take your exams very seriously because it's not a time for me to joke around. I'm very serious when it comes to your exam preparation. And as much as I am serious about your exam preparation, I hope you are serious about your exam preparation. Prarthana, it's okay. I hope you're coming back and watching the recorded bit of it, right? Diagrams for fundamental unit of life. Look at all the... try to have a look at the cell diagram in general because they may ask you cell diagram. Nucleus ka diagram, look at, I don't have the textbook with me, but these are some broad things which are there at the top of my mind. I'll make a short and I'll tell you which are all important. More than drawing, diagram based questions come. Which is why the kind of questions we solved is very important. Ma'am, if cell will shrink, no ma'am, but in image A is smaller than B. I'll go back. A is not smaller. See, that is it's a hypothetical one. They'll not give you exact one. But what we need to focus on is this part, right? So here they're just trying to show you that the protoplasm content has shrunk. Yes? Ma'am, these questions will come in exam. See, I hope that exact question comes so that you all will get, 20 out of 20 or 25 out of 25. But similar type of questions will come. Is it necessary that only plant cell has cell wall? That is one good question. They can ask you in the exam as a part of competency. Why is it that only plant cells, and I want you all to give me the answer. Why is it that only plant cells have cell wall? Why not animal cells? Why not animals? I have covered this particular topic in my video. Why not animals, right? Diagram is tricky. For protection, okay. They are exactly Hema and many others like Prisha, Avni, Abhimanyu, Ananya. Yes, plants can't dance and move around us. Exactly. See, in our case, we are able to physically protect ourselves. See, today it's very cold outside, or for example, if it's very windy, right? Like very windy, and I want to protect myself. I can walk and come and protect myself. But in the case of plant cells, if it's windy, if it is raining or whatever is happening, they can't go anywhere. They don't have umbrella and they're not waiting. No, rain is coming, I'll keep umbrella. Oh, it's very hot, I'll put one, maybe my sunglasses and just, chill around. Plants can't do that, right? Plants are where they are stationary, which is why in this particular case, we see that it's very important that they are able to protect themselves. When you say mechanical injury, it means, for example, say if a leaf falls down, or maybe, there is strong winds that are there, right, which can cause physical damage, right? So we're talking about physical damages to the cell that could happen if the cell wall wasn't there, right? Yes. Let's move on to round 3, everybody. Round 3 is going to be based on your practicals. Okay, now you have practical exams where staining of plant cells, you have some amount, it's there even in your NCRT textbook also. So this right here is going to be activity based. Yes, and in biology, especially for 9th and 10th, activity based is very, very important, right? So let's get started. Before scraping the inner side of your cheek to prepare a temporary mount of cheek cell, the mouth has to be rinsed. Why? Right? To remove food particles, to make it alkaline, to avoid injury, or to make it acidic. Don't overthink. I only have this. Please, my dear students, I am giving you a hint because I am a Leucosaurus. I request you sincerely not to overthink this. Why will you wash your mouth? You think about it quite reasonably. Everybody is telling me to avoid injury. See, we are talking about the mouth being rinsed when you are preparing a cheek cell. Now see, think logically. Where can you answer? Live chat page. See, when you say acidic and alkaline, it is with respect to the pH, which of course you will be learning in chemistry. Kriti ma'am will be telling you. But basically pH is with respect to how much of hydrogen ions are going to be there, right? More number of hydrogen ions, that means more acidic. More number of hydroxyl ions or lesser number of hydrogen, we know that it's more alkaline. So alkaline is a nature of the thing, right? It's to remove food particles. See, when you're going to take a scraping of the inner cheek and you are going to put it in a slide, right? You imagine that there's a mount of cheek cells, but there are also tiny, tiny food particles. If you don't wash it and take the, sample, then these food particles that are there will interfere with your result, okay? It will interfere with your result, which is why in this particular case, you need to cleanse it, right? You need to cleanse your mouth so that you get a proper sample. I told you, don't overthink this. The answer was very direct. Okay? Next one. Ma'am, I'm not able to understand the question. So this is based on a experiment which is there in your textbook, right? How you can observe cheek cells under the microscope. For that they have put step by step. These questions are based on that, Prisha. I hope that all of you have your textbook, right? Now for those of you who are a little confused about this, see, basically what happens is that if I have to see cheek cell under the microscope, no, I need to take it's called a glass slide, right? I'll take a glass piece which is shaped in a rectangle, and in order for me to see it under the microscope, I need to get some of my cheek cells, no? So what they normally do is there's a device through which they will scrape it, okay? Inside aise dal ke, they will scrape it and then they will spread it. Now the question is asking, why should I wash my mouth before I do that? So that if food particles are there in my mouth, they will not interfere. Now question number 17, everybody. While observing an onion peel, right? So now here we are taking out an onion. Inside the peel, you take a thin layer and you put it in the slide, right? You take a thin layer and you will put it in the slide. Paheli noted its characteristics. Which among the following does she not see? Yeah, I know I leaked the answer, but still. Cells attached edge to edge without intercellular space, presence of single nucleus in the cell, presence of centrioles near the nucleus, or is it all of the above? We're talking about what is it that she does not observe under the microscope? Everybody's telling me C. Now I want you to tell me why. Why? Yes, because she can't see centriole. Okay. Give me why. I need an answer. If it's a 2-mark question, the why question will also be there, right? So they will ask you why for the same. Centrioles, exactly, right? So as we all know, centrioles is a structure which is not there in plants, right? Now see, onion peel ko, if you observe under the microscope, you see that they look like these long structures, right? So we are saying that if I had an onion, outside ka peel you take, so it looks something like this. The inner white part, you take one thin layer and you put it under in the microscope, right? You take that glass rectangle piece and then you put that here, put whatever stain and everything. Inside it will look something like this, okay? Now all the cells are attached edge to edge. That means they're all attached together, right? So all the cells, they will be tightly packed like this. And we see that there will be a dark structure which will look like this, which is the nucleus. But what you will not see is the centrioles, right? So centrioles is something which we will not see. Yes? Okay. Now next one is question number 18. Inter means between. Inter-house means what? Inter-house competition is between two houses, right? Intra means within. So if I say intracellular, within the cell. Inter means between two cells. Centrioles are structures which are present, right? They are small tubular structures which are there. They are enclosed by a, if they are enclosed, if the centrioles are enclosed, they form something called as centrosome. Little bit there, here and there. Yes. Sakshi and everyone others, I have given you the answer. Moving on, water gathers in the hollow portion of where? You have A, right? You have option A here which has only water. Option B here wherein inside we see that there is sugar solution. Alright. So outside you have water, inside you have sugar solution. Now in C, outside you have water, inside you have salt solution. Right? Then of course, as we all know, you have boiled potato and inside you have sugar solution. Now, where will water gather? That means they're saying in the hollow portion. So that means when I took my potato, right? I took a potato, I cut the top part, andar ka I took all my potato and I made a hole like this, right? I took all whatever is there. So this right here is what is hollow. That means there is a gap, right? So we see that there's a gap. Now I have kept a solution. Yes? Now that is what we are seeing differently. One may only water is there and the potato has nothing inside. That is in A. Second one may under there is water, right? Inside we see that there is sugar plus water, while outside there is only water. In the third one we see that there is salt, right? So in C we see that there is salt, while in the fourth one we see that there is boiled potato. Many of you have got the answer. Correct answer here is option number D, which is B and C. Now I'll take this opportunity to explain a very important concept, right? When we talk about solution, what is a solution, everybody? Very quickly in the live chat, can you tell me what is a solution? Now Kriti ma'am will be very proud to see that all of you have given me the correct answer, right? So what is a solution? Fatafat. Live chat pe. Very good, right? It's a homogeneous mixture. Yes, it's a homogeneous mixture which mainly includes a solute which gets dissolved in a solvent, right? So normally we see that solute particles can be solid, they can be liquid, but for our convenience, right, to understand this, we see that let's assume there is a solution which has a solid solute particle and we take it as a liquid. Now of course we have three kinds of solution, right? So we observe that we have three types of solution. So very quickly, all of you, what are the three types of solutions? Let's recall this concept because a lot of application based questions like the ones we discussed comes from this particular topic, right? So we see that we have hypotonic solution. So let me start differently. We see that we have isotonic solution, we have hypotonic solution, and we have hypertonic solution, right? Now we have three different kinds. Now what do we mean by hypo, hyper and iso? Now see, iso here means, assume that it is same. I'm not taking the literal meaning. Iso means same, right? So as we all know, if I had a beaker, right? And I had some solution, right? Let's assume there is some solution which has solute plus solvent, okay? And I put a cell inside. Now I'm putting a cell inside this. It just so happens that inside my cell also there is cytoplasm which has water, there are substances in it. Now basically inside also solute plus solvent is the same, right? So inside the cell is equal to outside, right? So if inside and outside are the same, will osmosis happen? Osmosis is movement of water molecules from region to higher and lower, right? So we see that the size of the cell will be the same because everything, there is no movement, yes? But what about in the case of hypo? Hypo means less, right? Now less of what? Less water, less solute, what is it? So imagine in the case, right, I'm going to remove this, alright, so I'm just going to make it easy for me. This I'm going to write S plus S. Now in the case of a hypotonic solution, right, what I see is that there is less solute. That means that if I have 1 liter of water, I have added only 1 gram of solute. That means so much of water is there but very little solute, right? But what does it have? More of solvent, right? So this less is with respect to solute. Now what do? I'm going to put a cell inside. Now because there is a difference, right? Cell pe what is there? We see that solute is more and solvent that is there with respect to my solution is less. So what is going to happen? Inside the cell we see that there is more amount, or you can say that outside the cell pe there is more amount of water than inside. So where will the water move? Yes, end osmosis happens, water will move inside. So what's going to happen to the cell? What is going to happen to the cell? Very quickly. Yes? What is going to happen, bachas? End osmosis, cell will swell. Exactly, cell will swell. Simple. Exact opposite happens in hypertonic. Okay? Hyper means, oh, Aishwarya ma'am is very hyper. That means what? She's full of enthusiasm, more enthusiasm, right? So at this point, what happens? So again, if I draw the same thing, see, I'm drawing it for you so that you can take a screenshot later, right? We see that there is more solute, and there is less solvent, right? While in the case of your cell, it is the ulta, right? So we see that there is more solvent, more, and solute that is there is less. I'm just drawing arrows. Because of this, what is going to happen? Water will go from outside, right? So we see that outside there is less of solvent than the inside. So water will move out, right? So when water moves out, what will happen? Cell will shrink. Yes? And exosmosis takes place. Students, quickly take a screenshot of this, right? Whoever has doubts in this particular topic, I want you to take a screenshot right now. Yes? Jaldi, fatafat, please take a screenshot so that you can write this down later. Yes? And let me know, because see, once I go out of the screen, you will not be, once I change the slide, you will not be able to take the screenshot. But many of you who had doubts, right? Whoever had doubts, are you all clear? Are we all clear with this? Yes or no? I need your answers, jaldi. Very good, very good. Okay. Make your own notes after this. Ma'am, does boiling kill or does it disrupt? It actually disrupts the whole cell. Very good. Moving on to question number 19, right? Question number 19, another 20 minutes pe we will wind this up, 10 more questions only, right? A cover slip is placed over the specimen to do what, right? Is it to prevent the tissue from drying? Is it to prevent glycerine from leaking out? To remove extra stain in water? Or to crush the cells? See, now what is a cover slip? Let's take the example, right? Cover slip is a small box, okay? It's a small, a rectangular box like thing which I will place. So imagine if this is my cell or whatever, my cheek cells that are there, I scraped and I put it on this rectangular glass. I put some drops of water, I put some stain so that I can see everything clearly. Now I will cover it with this, okay? On top of this, I am going to put a cover, right? And because I am covering it, I will call this as a cover slip. Now the question is, why am I putting this extra one? Why is it that we are putting an extra slip? I'll do this, right? I mean, I'll do this experiment. I feel like the sampling and everything is something I have to do, right? Lingesh Raj. So what do you do? Imagine, let's take onion peel. I took the inner peel of it, right? I took the inner peel and now I have taken my rectangular glass and on top of that I have put my peel, which will look something like this, okay? Outside it will look only like this. Under the microscope only you will be able to see the cells. Now I will add some drops of water. Okay, so I'm going to lightly shade. I'm going to add some drops of water. I would have added, maybe my stain. Then there is a cover slip, which is a rectangular box. Okay, we see that it's a rectangular box like this, which I will put on top of my water and my stain and everything. So why am I doing this? Yes, why am I putting that cover slip? Everybody, I think I would have missed your answer. So that air bubbles do not enter. Okay. We don't even have that as an option. To remove extra stain and water. But that I can do otherwise also. If I add the right amount of water, ideally there will be little water I will add, right? Ideally you should remove your extra stain and water before you put the cover slip. Otherwise air bubbles will come. What you should be doing is, once you stain it, get rid of excess water, put little drop of water and then put the cover slip. But why do I do that? Prevent glycerine from leaking out. Many of you are confused. See, correct answer is to prevent it from drying. Now what happens is that in order to observe it properly, right, we put the cover slip. Yes, so we need to make sure that in order to observe it, right, you need to make sure that you are able to see structures clearly. So when you put that little drop of water, you put it so that it does not dry and the cells are alive. Because sometimes what will happen is, if it is left without water for a long period of time, the cell can get dehydrated and it will not be proper, right? So in order to make sure that drying out doesn't happen, you ensure that you put the cover slip. Prevent glycerine from leaking out. See, again, it's all about, like, you add the cover slip, but that's not the main reason, right? For prevent it from leaking out. But why is it that you don't want it to leak out? That is the main reason, so that it doesn't dry, right? Which is why in this particular case, it is not to remove extra stain. If you crush the cell, what will you see? See, there are different ways of approaching it, okay? But my primary thing is, if you crush the cells and you will not be, cell organelles though in compound microscope, you will not be able to see clearly. You need a better focusing microscope, right? Which is why in this particular case, the correct answer here is option A. I hope all of you are clear with this. Yes? Are we all clear? Yes or no? Very good. Now, this is a slightly tricky question, okay? Now, this is again with respect to your activities, and now I understand that maybe we should do this activity together so that you understand what is what, because I know that this is also very relevant for your practical exam, which is going to come at the end of the year, right? So, either I will do it now or closer towards the end of the year, I will do it and I will show it to you, okay? Now, given below are 4 options, all right? So, let's have a look. Given below that you see are 4 steps in preparing a temporary mount. Now this mount is nothing but that glass slide that we've been talking about from that time, okay? Now identify the correct sequence of operations. How will you prepare that slide? See, if you have to do this experiment, what is it that you are going to do? I will show you with the option so that you can give me the final answer. Yes? I can see 4, 1, 3, 2. 4, 1, 2, 3. Okay, everybody has agreed that 4 is the first step. Do you want me to go to the previous slide so that you are able to see better? Yes. You guys go back and see it. 4, 1, 3, 2. Okay. Asta is also telling me 4, 1, 3, 2. Let's see. 4, 1, 2, 3, 4, 1, 3, 2. Let's see. 2 options make confusion is coming. What is it that you will do first? So we have understood that the first thing that we need to do is to rinse our mouth, right? So option number 4 is first. We need to rinse our mouth with fresh water, and we know we have to get rid of all the unwanted particles that are there that could interfere with my result. After that, what is it that I am going to do? What will I do? Will I add 2-3 drops or will I do glycerin? Right? So at this point, what I'm going to do is I'm going to take a scraping, right? So first I will take a scraping from the inner side and I will spread it, right? Then what is it? Oops, sorry. Then what is it that I will do? I will add my methylene blue. I will add my stain first, right? I'll add my stain. Then I'll make sure that I'll of course add it with a little bit of water also. And then in the end I will add glycerine also. Which is why in this case the correct answer that is there is 4132. Yes. So as you can see here, oh, I can see that there are two options with the same answer. Ma'am, B means, I know B, I'll give you marks because 4132, 4132, B and C are the same options. Yeah, B and C are the same. See, this is where you'll get extra marks in the exam. I'll give you marks. See, okay, I'll give you marks. Don't worry. B and C are the same, but most of you got the answer. I'll give it to you. 4132, if you've given me the answer, I'm fine with it. Okay? Marks are given to you. This is the point where I'm very happy that it's not menti. Because if it was menti, no, all of you be like, ma'am, my rank went because of you. But it's okay, right? At this point, I'm able to give you all marks. So at this point, all of you have got 20 out of 20. How are you feeling at this point, right? You've done with 20 questions and at this point we have revised so much, right? We've revised so much of the chapter. Now you understand that when you are revising, you always start from cell is fundamental unit of life. This is cell theory. Then this is plasma membrane. But now you see you're getting questions from different angles of it. So you are able to apply it, right? So that is what we want. And don't worry, right? Don't worry. We are going to make sure that we look into it at great detail. Don't worry, I know half an hour is there, 10 questions we will be able to do. So let's move on to round 4, everybody. Now round 4 that is there is assertion reason. See, assertion reason may you are all very pro. If you have attended my class very regularly, you guys are pro in assertion reason, right? So let's pick up the pace a little bit. Now assertion reason is endoplasmic reticulum lacks ribosomes. The endoplasmic reticulum that lacks ribosome is called smooth endoplasmic reticulum. SER is mainly involved in protein synthesis. What is the correct answer? Yes? Okay. See, if you are regular in my class, I always tell you in assertion reason. Now, I always reiterate this because there could be somebody new who's there in my class. There are 2 steps, okay? 2 steps you need to follow when it comes to assertion reason. Step number 1 is to just look at them as statements, okay? Just look at it as statement 1 and 2 and identify if they are true or false, right? Now, assertion statement A says that endoplasmic reticulum that does not have ribosome is called as smooth endoplasmic reticulum. We know that this right here is true. And the type of endoplasmic reticulum that has the ribosomes is called as rough endoplasmic reticulum, right? But is SCR involved in protein synthesis? If ribosome is absent, it cannot do protein synthesis. It does lipid synthesis. So this right here is false. Now, if this right here is false, you don't need to go to step number 2. Because step number 2 is applicable only if both statements are true. And in this particular case, both statements are not true. So you can eliminate A and B, right? Giving you the correct answer, which is option number C, right? Assertion statement is true, but reason is false. But say if both statements were true, then you will ask yourself if the Y statement justifies it. So we have further questions where we can apply the second part of the technique. Now let's move on to question number 2. Assertion statement says that cell wall is the non-living part of the cell. Reason being it offers protection, definite shape and support, right? So everybody, step number 1. What is step number 1? Identify if statements are true or false, right? Statement 1 says that it is non-living part of the cell. We know that this particular statement is true. Reason statement, it offers protection, definite shape and support. Very quickly everybody, is this statement true or false, right? The statement is true. It provides a definite shape, it provides rigidity. Now we are at a condition where we can eliminate C and D, right? Where it's saying one among the 2 are false. We can eliminate this. Now we go on to step number 2, right? Now we have to see step number 2. Now step number 2 is to identify if this is the correct reason. So how do you identify if it is the correct reason? You will ask why after the assertion. Just read it and say why. Cell wall is a non-living part of the cell. Why? It offers protection, definite shape and support. Can we say that that's a proper reason? Is it justifying my why question? Yes or no? Is it justifying my why question? I want a yes or no. It's non-living because it provides it, or is it non-living because it is made up of cellulose? It is non-living because it is made up of cellulose, right? And because it is made up of cellulose, it's giving this. But it's not non-living because it provides rigidity, right? That's the function. Which is why in this case, the correct answer here is option B. So many of you struggle with identifying this particular point, which is why I've spent some time here, right? So that in the exam also you ask yourself this very question and you reason yourself out to identify it, okay? Very good. This technique works across physics, it works across chemistry, it works across social studies, biology. You can write for mathematics. I don't know if it works in mathematics, but see, assertion reason ka approach is the same, right? So I'm telling you it works across subjects. Trust me, it's going to be very simple and easy. Yes? Now moving on. A cell swells up when present in a hypotonic solution. Reason being that more water molecules will enter than they leave, right? Yes. See? Very simple and easy. A cell swells up, right? When present in a hypotonic solution. True. Reason says that more water molecules enter than they leave. That is a continuation of the statement, which is also true. Now that means your C and D is no longer there. Ask yourself, why does a cell swell up when placed in hypotonic solution? Because of the movement of water, right? And more water molecules enter into the cell than they leave the cell. Which is why the correct answer here is option A, that both statements are true and R is the correct explanation for A, right? So now you are able to get the understanding. Now students, very quickly before I go ahead, how many of you here are now feeling confident about this? Arey ma'am, assertion reason though we will be able to manage. You're feeling confident that tomorrow if you get an assertion reason question in your exam, right, you will be able to manage, right? That's all that I want. Confidence is key. Half the time in exams, if you have confidence, no, trust me, you will get marks, okay? And after you're done with science exam, you come and tell me how it was in the chat, okay? Yes. Samajh mein aa gaya. Yay, I'm also so happy. All right, let's go on. I think Seema had a doubt. Let me know in the comments, Seema. Seema, it's okay. Don't say doubt. You asked me the doubt. It's okay. Next one is for you, right? I'm not going to teach you the approach. Now you have to give me the answer, right? Assertion says that prokaryotic cells are primitive and larger than eukaryotic cells. Reason being that prokaryotic cells lack cytoplasmic membrane-bound organelles. Seema, I've told you technique, okay? I know assertion reason mein problem hota hai, but I've told you technique. Now you have to apply. No, it's like maths. Formula hota hai, you have to apply formula and then you have to try to do it yourself. If you make errors, it's okay. But you should not be like, ma'am, I'm not trying. I've told you the technique. If you have not understood, rewind back and watch it. Yes? Very good, all of you. Very good. Prokaryotic cells are larger than eukaryotic cell. Is bacteria larger than an animal cell? No, this statement is false. But do prokaryotic cells lack membrane-bound cell organelles? Absolutely true, right? Let's go on to the next one. Assertion statement says that mitochondria are known as the powerhouse of the cell. Reason being they produce energy as glucose for various cellular activities. What is the answer to this particular question? Mitochondria is known as powerhouse of the cell. Absolutely, right? It is true. But they produce energy as glucose. Yes or no? Do they produce energy as glucose? See, they've put in the bracket. No, they don't produce energy as glucose. Rather, they utilize glucose, but energy is produced in the form of ATP. Glucose undergoes breakdown, right? Complete breakdown of glucose in the presence of oxygen in order to release energy in the form of energy currency, that is ATP, right? Which is why the reason here is false. Students, I know glucose is the raw material. It is not the end product, right? So correct answer here is option C. Mitochondria is powerhouse of the cell. True. Do they produce energy in the form of glucose? No, in the form of ATP, right? So with this everybody, we move on to last round of questions, okay? Round number 5. Now round number 5, I think we have about half an hour. 5 more questions, okay? 5 more questions, and let me tell you, these are competency based questions, okay? They are competency based, which means little bit of application is there. Does this mean that you guys are going to get scared? Are you going to get scared? Yes or no, ma'am? Competency means I can't do. Are you guys going to get scared about it? You stayed here for 1.5 hours and you've understood everything that you need to know about solving questions. See, I told you today was not going to be about me teaching you the concept. I have done that for almost 1.5 months. Ankita ma'am and I have done it together. Today is about you guys applying your knowledge, building confidence to know that you've studied and worked hard enough to know that you're good enough, right? You need to tell yourself that. You need to build confidence, and we are here to do that. So none of you are going to be scared about all this competency, HOTS and all this we'll come up with, or your teacher will tell you, but nothing to get afraid about because you have that confidence, right? So let's get started with question number 26. Which among these properties, I feel like I've built it up overly, but this question is very easy. But which of these properties qualifies amoeba as a eukaryote, right? It is unicellular, it needs food for energy, it has a membrane-bound nucleus, it is surrounded by plasma membrane. What or which among these properties is actually qualifying amoeba as a eukaryote? U means what? U means true. What does carry-on mean? Carry-on means nucleus, right? So true nucleus. So if I have to say a cell is eukaryotic, it is with respect to the fact that it has a membrane-bound nucleus, right? So correct answer here is option number C. Very good, all of you. Very, very good. Now let's move on to a little bit on cell division. I know this is going to take some time. Now this is a hypothetical cell. We are not saying this is an animal cell, plant cell, amoeba, nothing. Okay? We're just saying that there is a cell. Okay? And cells grow or they increase in number by dividing. And here there's a picture shown of a dividing cell where you see that there are some chromosomes. Okay? How many chromosomes are you able to see? Yes, how many sets that are there? You are able to see 4, right? Now your question is, at the end of cell division, how many chromosomes will each daughter cell get, right? We're talking about cells growing by dividing. So we're talking about with respect to growth. So with respect to growth, which type of cell division takes place? Does equational division happen or reductional division? Now we know that with respect to growth, repair and all of that, it is equational division or it is mitotic division or mitosis, right? So in all the cells except your reproductive cells, we observe mitosis, which means that if my parent cell has 4 chromosomes, how many chromosomes will my daughter cell get? It will also get 4, right? So correct answer here is option B. Now the question is, the next part of the same question. How many cells are formed after cell division? Okay. Now first you have to identify how many chromosomes will go. Hypothetically, there is a parent cell having 4 chromosomes, right? Now they are made duplicate copies, right? Because you know that cell cannot become half, where it has half the DNA, half the number of cytoplasm, half the number of mitochondria. Cell cannot function. So it will make a whole copy of all its DNA, all the organelles, all the cytoplasm, and then they will divide. How many cells are you going to get at the end of it? We're talking about end of mitosis. You will get 2, right? So you will get 2 daughter cells. Now imagine if this was a reproductive cell, right? If this was a reproductive cell, at the end of cell division, how many cells are we going to get? If it was a reproductive cell, how many will we get? Ma'am, when parents have 4, how can 2 daughters get 4? See, I hope you watched the session on cell division because I did that a little detail. It makes it easy for me with props. You have a parent cell, okay? What the parent cell will do, it will make a Xerox of everything it has within its body. Okay, it will make a photocopy of everything. Okay, it will copy, it will duplicate. So the Xerox in biological term, we will call it as duplicate, right? So it will duplicate everything within its body, within a cell. Then what will happen? So ideally there were 4 chromosomes. It made one more copy, and we know that chromosome is actually like this. But we know that during cell division it gets this X-shaped structure. Now what eventually happens is that this will break, so that one cell will get this, the other cell will get this. Yes? So at this point, if there are 4 such X-shaped structures, they will all divide, right? So we see that when this will split into 2, 4 will go here, 4 will go here, eventually giving you 2 cells, right? See, cell division may, I have done a class, Sumit and many others. I have done a detailed session on this, right? So let me tell you that you go watch that particular class so that it will help you out, okay? Because I think the fundamentals are not clear, okay? No, nobody, please don't tell me that, ma'am, I have stupid question. I will never ever entertain that. Every question is a relevant question, but like I said, I may not be able to go into the details of it because of time constraint, but my main intent is that whatever conceptually you are not clear, I want you to focus. Tamanna, Smriti and many of my students, please pay attention in class. Ma'am, how 4? In reproductive cells, meiosis happens, right? So 2 steps are there of reductional division, so you get 4 in reproductive cell. But in mitotic, you get 2. Alright, so let's get started. So this is just the question that is there in the next slide. So let me just go there. Now as you all can see, we have a diagram of the animal cell, right? So you see that you have an animal cell and you need to identify the part which is labeled as X. Part which is labeled as X. Is it nucleus? Is it chromosome? Is it Golgi? Or is it endoplasmic reticulum? It is pointing towards this one here. There is another flattened. See, I'm leaking. But see, you have another flattened structure here. Easy peasy. Exactly. Right? So the correct answer here is option D, which is endoplasmic reticulum. Very good, all of you. Very good. Now students, just before I go ahead, right, just before I go ahead, I can see that good number of you are here in the live class. I really hope that all of you have hit the like button. If in case you came in late and you've not hit the like button on this video, now is the time for you to hit the like button and subscribe button, right? Yes. Ma'am, 4 chromosomes, then how 2 cells? But in cell division, what is happening? Parent cell is dividing into 2 halves, right? Which is why 2 cells may 1 set of chromosome here, 1 set of chromosome here. Yes? Hope I have clarified. Moving on to last question, which is not an MCQ, but we just added it here. Now the question is, which cell organelles are found only in a plant cell that are not shown in the diagram? You need to write yes or no for each row, right? So you have to say if cell wall is present, yes or no. Ribosome is present, yes or no. Chloroplast is present, yes or no. Shrivali, I'll do it in the end of the class. Features found only in plant cell. Read the question. Cell wall, yes. We find it only in plant cell. But ribosomes will I find only in plant cell? Hey, who is telling no? Who is telling no for cell wall? Everyone. 8th standard say you've been learning cell wall is there in plant cell. Okay. Now I will feel bad if you say that there is no cell wall in plant cell. Okay. So of course with this everybody we know that ribosomes are not present, right? Ribosomes are not present only. See, understand that they are not saying that it is not present. No, we're not saying that ribosomes are not present in plant cell. The question is which is exclusively, that means only in plant cell. Yes? So I don't want to give that reaction, but yeah, for a brief minute I felt that way. But chloroplast, yes, we find chloroplast only in plant cell, right? Oh, what is this? Features found? I think it's the other way around. Cell organelles found only in which are not shown in the diagram. Oh, my bad. It's okay, I changed the question, but you are all right. The question was asking which cells are found only in plant cells that are not shown in the diagram. So it is ulta. No, yes, no. But it's okay, you are all right. I am giving you marks, right? I am giving you marks. I though changed the question, but it's okay. For my question, you all give me the correct answer. Yes? Okay. For my question, you are all right. Now, the question is asking which is not shown in the diagram, right? That is my main thing. Your answer in your parallel world is correct. So at this point, what has happened? All of you have got 30 out of 30. And here I'm just going to twist it and change it, right? That's why, ma'am, I got confused. Oh, I'm so sorry. My bad. My bad. It's my bad. So this is no, yes, no, right? No, because it is not shown in the diagram, but it's seen only in plant cell. Yeah, I know. It's exactly like that. Okay. So with this, everybody, yes. Ma'am, it was terrible. Kya hua, Shaurya? Is it that you're not feeling confident? Is that what happened? Or did you get confused in my explanation? That's what I'm a little bit worried about. But you tell me, what happened? Yes? Kaju Katli. Chalo. Ankita ma'am is better at this, but Kaju Katli for all of you for getting 30 out of 30 in this particular test, right? I will share the PDF right away, or maybe because I have a 7 p.m. class with your seniors, I may finish that class and then only send it. So please bear with me, right? Okay. Ma'am, I felt guilty when you said it's incorrect. Arey, my bad, no? I'm very sorry. I'm also human, right? I make some mistakes. Yes. Okay. So with this, everybody, I'm going to sign off. But before that, I'm going to take a minute to help your friends with this, right? So as you all know, when you talk about meiosis, okay, I'm going to take one question. Meiosis, that is, there is a type of cell division that we exclusively see in the case of reproductive cells, right? So reproductive cells, especially for the formation of gametes, yes? So we know gametes are the reproductive cells like your sperms and egg, right? And we know that here, especially in sexual reproduction, it's important so that to maintain the chromosomal number, right? Now, of course, we know that in the case of meiosis, there will be a parent cell, let's say, which has 46 chromosomes, like in our case. But in meiosis, what happens is that there is reductional division that takes place at 2 stages. So, stage 1, which we call as meiosis 1, is where the chromosome number becomes half, right? So 46 will become 23 and 23. But the division does not stop here. It will further divide in phase 2 or stage number 2, which we call as meiosis 2, where it will give rise to 4, or each of those cells will give rise to 2 daughter cells. And we see that this meiosis 2 that is there is like a mitosis division or equational division where chromosome number is the same. So you get 23, 23, 23. So overall, because meiosis number, the reductional division takes place, we call it as reduction. Because the reduction in number of chromosome takes place, we call it as reductional division. Now, why should number of chromosomes reduce? What is the whole point? Because in sexual reproduction, we know that there is fusion of gametes. Okay, here they'll come back and original number will come when zygote formation happens, right? So, which is why we see that this right here is important. 4 differences between plant and animal. Plant may you have cell wall, you have chloroplast, you have very big vacuole, right? And then you also know that in the case of plant cells, you can talk about how they have the ability to produce their own food structurally. Yeah, big vacuole. 4th one, I'm not able to think about, right? While in the other one, you can simply talk about it, right? And then there is, we see, no centrioles, no centrosome in plant cell. Other one mein centrosome is there, no cell wall, no plastate, small vacuoles or no vacuoles at all, right? Ribosome, small structures which synthesize proteins. Yes? So with this, everybody, you have a quick homework question. You are not done. If I let you go like this, then I would be a bad teacher. So homework question for everybody, where in the comments of this video, you will have to tell me the structure and function of the plasma membrane. This is a subjective question which you will have to write for 2 marks. Okay. So 1 mark I will give you for structure of plasma membrane and function for another 1 mark. Right? So I will let you know and I will be checking your answers, okay, in the comments. You know I am very, very active on the channel and I reply to all of you. So it's very important that all of you let me know. And further doubts that you have, let me know in the comments of this video as well. And as you all know, BYJU'S 9th and 10th has got you covered, right? So please make sure you don't forget to tell your friends about this and share this video with them. Do not forget to hit the like button and of course do not forget to hit the subscribe button because we have a lot more exciting things coming your way, right? So you You will get the notes of this particular channel in the Telegram channel. The description, the link to the Telegram channel is there in the description. But thank you so much, everyone. I see that a lot of you here have doubts. In case if I've still not covered your doubts, let me know in the comments of this video. And with this, everybody, I'm going to be signing off. I will see you all soon. But up until then, take care, lots of love, and bye-bye.